So the next speaker is Kathleen Green. Please, can you come on the stage? So Professor Green is an American cell biologist whose main focus is cell-cell communication. And she's professor in the Department of Pathology and in the Department of Dermatology at Northwestern University in Chicago. She also serves as Associate Director of Basic Sciences in the Cancer Center of Northwestern University. She received her PhD in Cell and Development Biology from Washington University in St. Louis, Missouri, and carried out postdoctoral training in the Department of Cell Biology in Chicago, where she joined the faculty a long time ago in 1987. She was also a visiting professor in the University of Cologne in Germany in 2015, and her main research focuses on how cadherins and their associated protein-direct tissue morphogenesis and differentiation. And her pioneering work on desmosomes contributed to the discovery of their involvement in disease of the skin, but also in the heart. She served as the secretary of the American Society for Cell Biology. She's past president of the American Society of Investigative Dermatology. She's an honorary member of the Society for Investigative Dermatology and for the ESDR. And she was also elected into the German National Academy of Science in 2016. She worked also in journals, so she's associate editor of Science Advances, deputy editor of Journal of Investigative Dermatology, and deputy editor-in-chief of the Journal of Cell Science. And finally, I'm very happy to see you in person because I'm more interested in hemidesmosomes than in desmosome, and I have never had the opportunity to meet you, but probably the gap between hemidesmosome and desmosome was too important, and now we are narrowing it, and it will become probably closer and closer.
Yes.
So you will speak about linking epidermal morphogenesis and skin inflammation through desmosomes. Please.
Thank you very much for that wonderful introduction. And I'd like to make the point that all junctions, cell substrates and intercellular junctions, are networked into something that cannot be uncoupled. So they work together. So hemidesmosomes and desmosomes included. Well, thanks so much. This has been really wonderful. I'm looking forward to meeting a number of you. There are a lot of potential new colleagues and also junior scientists that I'm really looking forward to meeting and interacting with at this meeting. So today's story is about how cell communication and cell communication mechanisms that evolved, that appeared during evolution and brought cells together, took on new functions to help us meet the challenges of living life on land. And it's a story about cell-cell adhesion molecules called cadherins. Now, these cell-cell adhesion molecules appeared first in primitive form in single-celled organisms. And someone might ask, well, why were cadherins in single-celled organisms? Well, it's been speculated that they may have been involved in capturing prey, and then later on, they were utilized by these multicellular wannabes, ancient organisms like sponges that were creating multicellular sheets. These cadherins brought those cells together, but they also acted as an opportunity to anchor the cytoskeleton to the plasma membrane. And in these ancient organisms, the cytoskeleton was actin cytoskeleton. We haven't seen intermediate filaments yet. Then later in vertebrates, as the organisms and tissues within them became more complex, we see the emergence of a new type of intercellular junction called desmosomes and their associated desmosomal cadherins. And those cadherins reached their height of complexity in terrestrial vertebrates, where they expanded along with their associated keratin intermediate filament proteins. And the expansion, and they took on new functions that superseded their role in cell-cell adhesion and gave our tissues new properties that helped us build new, better barriers. Now, we see here a very important illustration of the textbook function of desmosomes, and that is intercellular adhesion and mechanical integrity. So what we've done here is uncouple the intermediate filament cytoskeleton from desmosomes by expressing a mutant of a protein called desmoplakin, which is an anchoring protein that anchors the intermediate filaments to the plasma membrane of desmosomes. And if you do that uncoupling, what happens is that these tissues fall apart very easily under mechanical stress. And desmosomes are found prominently in epithelia, such as the epidermis, which is the focus of today, of course, but also other tissues such as cardiac tissue, the heart. And in all of those cases, if you interfere with desmosome function, you get a number of types of disorders. In the epidermis, those disorders range from fairly mild stripe, palmoplantar keratoderma, for instance, to lethal diseases like epidermolysis bullosa, a lethal form of that disorder. And I'll be talking about inflammatory disorders today that mimic, in a way, psoriasis, common disorder that we just heard about from Nico. And in cardiac tissue, if you interfere with desmosomes, it leads to a number of types of disorders, primarily something called a rhythmogenic cardiomyopathy, which can lead to sudden death, particularly in well-exercised individuals. So we all know very well that the epidermis is a regenerating tissue. It regenerates about every 28 days through a process of proliferation of cells in the basal layer, and this, of course, is where the stem cells or progenitor cells reside. And then those cells commit to differentiate, and they transit into the superficial layers where they undergo a very well-programmed process of differentiation that I think we're all pretty well familiar with, and ultimately the creation of the life-preserving cornified envelope. And of course, this epidermal barrier provides multiple functional layers that provide a physical barrier, protect from water loss. They provide ultraviolet radiation protection, and of course, they're an important immune barrier. And in my talk today, I'm going to talk about how the role of desmosomes, which play a critical role in every step of this epidermal differentiation process, how desmosomes actually link the formation of multiple functional layers, the multicellular barrier, with the immune barrier through a shared mechanism that I'll introduce to you today. So now I want to take a step back to our cell biology courses just to make sure we're all on the same page. I think most of you are familiar with these intercellular junctions that are present in, and I'm showing you here, in simple epithelia, just to start out with a simpler situation. There are a number of types of intercellular junctions, and I'm sorry I don't have hemidesmosomes on this slide. I only have the, they're there, they're not labeled. But yes, the hemidesmosomes, very important, are down there. The intercellular junctions come in three types here. The tight junctions at the most apical region of the lateral membranes between the cells that provide barrier functions. They keep proteins in the right place. And then we have our adherens junctions. These are the more ancient cadherin-based junctions, and the desmosomes along the rest of the lateral border. And these adherens junctions and desmosomes, both of which anchor different cytoskeletal elements of the plasma membrane, have similar blueprints with the transcellular cadherins. Desmosomes are a little more complex. I think all of you in this audience know there are a couple of types of desmosomal cadherins called desmoglians and desmocollins, which work together in ways we're still trying to understand to mediate adhesion. And they also act as a scaffolding for cytoplasmic proteins in the armadillo family. Very important family I won't be talking about today. We're all familiar with beta-catenin and Wnt signaling. Desmosomes have their own armadillo family members. And then, of course, there are the cytoskeletal linking proteins. Alpha-catenin, which links to actin, and in desmosomes, desmoplakin, which is an essential protein found in all desmosomes that anchors intermediate filaments. Now, I alluded a little bit at the very beginning before I started my talk that all intercellular and cell substrate junctions really are networked both physically and functionally. And this is very true for desmosomes and adherens junctions. They're physically and functionally linked. And important later in my talk is the idea that desmosomes balance the contractile signaling forces generated by adherens junctions and can even affect the ability of adherens junctions to recruit things like vinculin, which strengthen adhesion through the actin network. Okay, so this is a simple situation, relatively simple, but when we get to the epidermis, we have multiple layers, and in each of those layers, desmosomes have a different composition. And this is due to the fact that there are a number of different desmosomal coherent genes, and they're encoded proteins, and they are present in a patterned layer throughout the apical to basal aspect of the epidermis. So the composition of desmosomes is different in every layer. And our hypothesis driving a lot of the work in our laboratory is that overlaying this polarized desmosomal coherent pattern on top of the more ancient actin classic adherent network provided tissues with the opportunity to develop more complex functions. And for the rest of the talk, I'm going to focus on one of these desmosomal coherents called desmogliin 1, which is only present in stratified tissues of terrestrial or previously terrestrial vertebrates. It's expressed first in cells in the basal layer that are beginning to commit to differentiate, transit into the superficial layers in the process of stratification, and then is present at its highest levels in the superficial layers where tight junctions are present, and is important, in fact, for tight junction assembly and maintenance in the epidermis, a subject that I won't be talking about today. But desmosomes are also present in keratinocytes. As I mentioned, they're present keratinocytes. They hold keratinocytes together, but within a more complex collection of cells within the epidermis. And we all appreciate that keratinocytes, although they're the most abundant cell, are present among a number of other types of cells, including dendritic cells, Langerhans cells, melanocytes, and immune cells. And those keratinocytes are constantly in crosstalk with those cells. Now, desmogliin 1, as I mentioned, is in this patterned layer. It's expressed first in the basal layer, but at its highest level of expression in the superficial layers. And this is important because it has both suprabasal, basal, and suprabasal functions. And in fact, up here in the suprabasal layer, it's well positioned to actually respond to environmental stress of a variety of sources. And in fact, we've demonstrated that ultraviolet radiation and other things like bacterial peptidoglycans cause the temporary acute loss of desmogliin 1. And this is actually protective because, as J.J. Johnson and Chris Arnett showed, temporary loss of desmogliin 1 actually results in paracrine signaling from keratinocytes to melanocytes to increase pigmentation. But chronic loss of desmogliin 1, which occurs, for instance, in certain patients, like patients with SAM syndrome, which is severe dermatitis, multiple allergies, and metabolic wasting, leads to an inflammatory disorder with features that resemble psoriasis. So acute loss is a protective, but chronic loss, we think, is pathogenic. And what I'm going to do for the rest of the talk is tell you a story about how desmogliin and desmosomes function in helping to create or develop the epidermis actually shares mechanisms that are immune or inflammatory suppressive mechanisms. Now, how could this be the case? Well, I'm going to try to convince you that an actin remodeling complex regulated by desmosomes links these two different, very important functions in the epidermis. So a model you use that I want to introduce to you are 3D organotypic skin cultures, which many of you may be familiar with, which are created by taking cells from humans, putting them in culture on a fibroblast feeder layer, fibroblast collagen feeder layer, and then lifting those to an air-medium interface, and this triggers the stratification and differentiation and something that looks remarkably like human epidermis. This is a very easily manipulable system, so you can put in retroviruses, lentiviruses, et cetera, to genetically modify these cultures. And a number of years ago, Spiro Getzios and Corey Simpson in the lab introduced a retrovirus expressing an shRNA to knock down desmogliin 1 as these cultures were developing. And what they demonstrated is that compared to the wild type, or the control over here, the desmogliin deficient cultures exhibited defects in the epidermal differentiation process, both the structural features, but also they were unable to express certain differentiation-specific keratins, for instance, like K1 and K10. They went on to demonstrate that this role of desmoglein-1 was through actually regulating ERB family member signaling. So in the basal layer, desmoglein-1, when it's expressed, normally inhibits EGF receptor signaling, and it does so through an associated protein that we found through a screen called ERB2 interacting protein. But in the superficial layers, desmoglein-1 actually supports ERB2 signaling, which is important for maintaining the tight junctions and the mechanical gradient which exists in the epidermis. So it plays very important roles in growth factor signaling, but also in regulating the mechanics of the epidermis. But beyond that, at the time we were looking at these desmoglein deficient cultures, we also noted that they exhibited changes, aberrant cell shapes, changes in the cell size, and importantly, we saw that unlike the control cultures, which have these what look like umbrella or mushroom-shaped cells, which are delaminating cells, and we know that delamination contributes to the process of stratification, in the desmoglein deficient cells, those delaminating cells did not appear. Instead, there were a jumble of cells that appeared to be being pushed up in order to mediate this stratification process. So a postdoc in my lab, Oksana Nekrasova, hypothesized that perhaps loss of desmoglein-1 leads to aberrant transit of those cells into the superficial layers and might actually be involved in stratification. And she carried out loss and gain of function experiments to address that possibility. And I want to show you the gain of function experiment because I think it is so surprising and striking. So what Oksana hypothesized is that, well, desmoglein-1 is only present in stratified tissue. So what happens if you introduce it into simple epithelial cells? Can you cause them to stratify? And so what she did is she introduced desmoglein-1 into simple MDCK cells. These are canine kidney cells, often used in cell biology of polarized cells and experiments with polarized cells. And what we can see over here on the left in these GFP-expressing control cultures is that the cells are dividing normally. Every once in a while you see one extruding. But over here on the right we see in cells expressing a desmoglein GFP fusion protein, these clusters, grape-like clusters of cells that are popping up or extruding in the process of delamination from the simple epithelial monolayer. And if you look in cross-section, you're actually seeing these cells stick to the surface of the monolayer and form another layer. Normally in simple epithelial cells, the process of apoptotic extrusion leads to the loss of those cells from the monolayer to keep the number of cells in those monolayers constant. But here, a whole other layer is forming. So this was really striking to us, and it suggests that a single molecule found in stratified tissues is sufficient to drive stratification. Now, it requires the existing actin cytoskeleton, other proteins that we identified, but it in and of itself can cause that stratification process. So what's the in vivo relevance of this in vitro observation? Well, to address that, we developed a desmoglein-1 knockout animal. That animal is actually perinatal lethal because ultimately the skin falls off because desmoglein-1 is important for adhesion as well. Now, but if we look during development between embryonic stage, about embryonic stage 13 to 15, when desmoglein-1 is normally expressed, we actually saw that if you measure the thickness of the epidermis, that at those critical times the epidermis is thinner. Later on, it catches up through some compensatory process that we're now analyzing through single-cell seq analysis. Now, I just wanted to show you a little bit of that single-cell seq analysis because it raises an important, makes an important observation that's important for the mechanisms that desmoglein uses to drive stratification, and that is during this time period, there is a downregulation of a whole signature, an actin signature driven by serum response factor, which is a transcription factor that drives actin signaling. One of those is this protein vinculin, which is very highly significantly downregulated. And to us, this suggested that somehow the loss of desmoglein-1 is associated with downregulation of the whole actin signaling process. Why is this important? Well, I'm going to tell you why it's important by asking the question, how does desmoglein 1 support stratification? Well, we took a page out of the notebook of people who were studying simple epithelial cell extrusion. And those people, both Jody Rosenblatt and Sarah Vikstrom's laboratory, looking at epidermal stratification, demonstrated that you require cells that are going to stratify and delaminate to become mechanically different from the neighboring cells in a process that is breaking the symmetry of the plane of the epithelial sheet. And that process of symmetry breaking requires changes in the actin cytoskeleton. And so we hypothesized that when desmoglein 1 comes on, it is actually breaking symmetry between the cell and the neighboring cell that's not expressing it. And so desmoglein 1, we hypothesize, is breaking planar symmetry. And so we decided to look at the actin cytoskeleton, and when we looked in these small differentiating colonies, we saw that colonies lacking desmoglein 1 had an actin cytoskeletal pattern. If you look here, we're thresholding here for the cortical actin cytoskeleton. And we can see compared to colonies that do express desmoglein 1, they lack the nicely organized cortical actin, but cells that do have desmoglein 1 have very robust cortical actin cytoskeleton. We went on to demonstrate that desmoglein 1 supports this actin organization by recruiting this complex of proteins to the cell surface. Now recall that desmoglians and desmosomes are thought to be anchors for intermediate filaments, so this is a non-canonical, previously unappreciated function of recruiting actin remodeling complex. This complex, which contains ARP2/3, has been shown to be important for polymerizing actin, and in this case we showed that it drives the polymerization of actin next to desmosomes, and this is important for breaking mechanical signaling, symmetry. Now, one of the proteins that's in this complex is something that we recently identified to be associated with desmoglein 1, and it's called NIC1. NIC1 is recruited to the desmosomes through this tyrosine 1042. Now, if you mutate that tyrosine 1042 to disrupt the interaction with desmoglein 1 and prevent the actin remodeling complex from being recruited to desmosomes, what we demonstrated is that it blocks this stratification process. So coupling of the actin remodeling complex to desmosomes is important for stratification. Okay, so that's summarizing what we know now about desmoglein promoting this multi-layering. What does that have to do with the immune barrier that I mentioned to you previously? Is there a connection? Well, in these animals that I demonstrated have this differentiation defect, we also demonstrated by RNA-seq analysis that they have an inflammatory signature. A signature that demonstrates, even before they're born, that they're susceptible to inflammation. This inflammatory signature actually resembles that exhibited by patients with psoriasis, so it's a Th17 IL-23 response. We also demonstrated that these patients that I introduced to you earlier, which have something called SAM syndrome, and also have these inflammatory rashes that appear like psoriasis, also exhibit a similar Th17 IL-23 response. Furthermore, in collaboration with Ron Cohen-Brock, who did a 2-year fellowship in my lab, and now he's back in Israel, who has SAM syndrome patients, he demonstrated that the keratinocytes, that the epidermis and epidermal keratinocytes in these patients produce a lot of IL-23, and if you treat them with biologics targeting IL-23 like ustekinumab, they get much better. And you can see that at the RNA-seq level, comparing before and after, that the Th17 signature is resolved and goes back to normal. So this suggests then that these, both in animals, mouse models, and in patients, that the loss of desmoglein 1, and this is the chronic loss of desmoglein 1, results in this inflammatory signature and phenotype. Now I'm going to go back and link the first part of the talk to this part of the talk and ask the question, can desmoglein-1 regulate inflammatory cytokines in a keratinocyte autonomous fashion? And is that actually linked somehow to this role in actin remodeling? So in order to address whether keratinocyte production of inflammatory cytokines is regulated by desmoglein-1, we set up an in vitro model system where we treated keratinocytes with TNF-alpha to induce IL-23 expression. And we demonstrated that this results in the robust expression of mRNAs for IL-23. And when we put desmoglein back into those cells, then we actually see a suppression of IL-23 expression. Now, there are no immune cells in these cultures, just stimulation by TNF-alpha, so this suggests that desmoglein-1 can suppress IL-23 expression. Now to ask, is there a connection with that actin mechanism? What we did is we put in this mutant protein, this protein of desmoglein that can't bind to NIC, which is the protein that recruits actin. And we saw that, lo and behold, desmoglein-1 with this single point mutation is unable to suppress IL-23 expression. So desmoglein-1 dependent dampening of IL-23 requires NIC1 dependent actin recruitment. How can this possibly be? What is the explanation for this? Well, it turns out that there was a paper published in 2018 which was, had created an animal model of psoriasis which knocked out a protein called NWASP. And you might recall that NWASP is part of this actin remodeling recruiting process. And so we wondered whether or not desmoglein-1 might be regulating NWASP and the distribution of this protein, which had been shown to suppress IL-23 in this animal model, the distribution between the cytoplasm and the nucleus. And so in order to address that, we did a fractionation, a cell fractionation, in keratinocytes that were and were not expressing desmoglein-1. And what we saw is a shift in the distribution of this important NWASP protein, such that cells that have desmoglein-1 have a lot of NWASP in the nucleus, and this NWASP has been shown to suppress IL-23. However, the loss of desmoglein-1 reduced the amount of NWASP in the nucleus, and what was left was a phosphorylated form that had been shown not to be able to suppress IL-23 expression. So this brings everything around into full circle, where we showed that desmoglein-1, by recruiting an actin remodeling complex to the plasma membrane, thus changing the mechanics of cells that are destined to delaminate, are also regulating the distribution of one of those actin remodeling complex, NWASP, between the cytoplasm and the nucleus. And we think our hypothesis is that this is responsible for suppressing IL-23, an important inflammatory cytokine in keratinocytes. And then just to finish off in this final slide, I just wanted to open it up a little bit because it turns out that inflammation has emerged as another potential common function of desmosomes in multiple tissues. Desmosomes in stratified epithelia, which I just told you about. In cardiac epithelia, there also appears to be an importance of desmosomes in regulating innate immunity, and the loss of this control leads to arrhythmogenic cardiomyopathy, or contributes to it, and which I didn't even, and one of the things I did not talk about today is gut, where desmosomes have been shown to also regulate innate immunity and other inflammatory pathways. So with that, I just wanted to thank everyone both in the lab at Northwestern University and also outside the institution who have really made a lot of this work possible. We couldn't have done it without you. And the Green Lab, former and current members, a terrific group. Shout out to Corinne Neeson, who my time in Cologne on sabbatical really was very stimulating and led to a number of these ideas. And finally, just wanted to make the last point that it's really important to stick together, both for our skin cells, but also in our scientific community. So thank you all very much, and if there's any time left for questions, I'm happy to answer them.
Thank you very much, Kathleen. Is there any question? I have a question about Desmosome. I'm wondering if you can have the same explanation between the communication between the desmosome and the nucleus and the induction of inflammatory cytokines for the same for Desmosome, because in, for example, in bullous pemphigoid, you can have induction of interleukin 4 or 13, and you have now clinical trial with for example, dupilumab in order to block this production of cytokine, and it's efficient. So I'm wondering if you can have the same induction of inflammatory cytokines, the same mechanism through actin.
Very well could be, and one of the things that I didn't talk about actually is that in these desmoglein 1 knockout situations, there is an impact on cell substrate adhesion. So we're actually also looking at possible changes in hemidesmosome proteins. Those changes could definitely contribute to what I showed was dependent on desmoglein 1. So if we look directly at the hemidesmosome, there very well could be a link. I'm not aware of anyone, it may be out there doing seq analysis on hemidesmosome deficient models. Maybe you're aware to look for some of these inflammatory pathways, but very well could be, yeah. And
And a more general question, because on the basal keratinocyte, you have at the same time desmosome and hemidesmosome, which is not the case later after differentiation. But just in this layer, could you imagine that you have a communication between both structures?
Yeah, absolutely. I mean, there are some cell substrate proteins that are actually at the lateral membranes. For instance, beta-1 integrin is at the membrane. I think BP180 as well, collagen 17, I think can also be sometimes at the lateral membrane. So I think there could be actually both direct interactions with proteins, but also through signaling and regulation of kinases and other things, I think that there could be indirect associations between those two. They're definitely networked functionally, but we still don't know a lot about that. It's a nice area for further investigation.
Okay, thank you. Thank you very much.
Okay.
Frédéric,
so, last question, Lionel Breton.
Yes, thank you, Kathleen, for you. I have just a question. During your presentation, you spoke about bacterial strain and pathogen on the global structure. Did you check the type of protease that could be really linked to the desmoglein and pathway?
The type of proteases that could be linked? Yeah. So both MMPs and ADAM proteases can, under certain circumstances, cleave off the extracellular domains. And of course, there's well-known, ETA is a well-known protease that can do it too. So there are substrates for a number of different proteases. So in terms of exactly what's happening under UV or peptidoglycan treatment, actually, I think Rich Gallo may have shown that an MMP can actually do it under certain bacterial exposure. But I think under each of these different situations, different proteases might be involved. So, but it is a substrate for a number of proteases.
Thank you very much.
Thanks.
Kathleen, so I will ask for the last speaker.